Okay, good afternoon. We are going to go ahead and start our lab today. We're going to take a look at the special senses. We're going to look at the eye and the structure of the ear, as well as the nose and the mouth. And all these slides are found in eLearn in your content section. I did rearrange them slightly. I moved all the slides on the ear up to just after the eye. You'll see what I'm talking about, because I don't want to get into any of the dissection slides today. So let's go ahead. You'll see what I'm talking about when we get there. Any questions on anything? Okay, then I'm going to open this up and go into the special senses. Everybody should see the slide. It says the special senses. Let me minimize this and bring that up. Okay, so everybody should see the slide. It says the special senses. When we're talking about the special senses, we're talking about the 5 specific special senses: smell, taste, vision, hearing, and balance or equilibrium. It's not touch. It's not pressure, it's pain. Pain, for some reason, is often included accidentally. Excuse me. Sneezing is not one of the special senses either. But people often mistake pain as a special sense, and it's not. It's only the smell, taste, vision, hearing, and equilibrium. They all require special sense organs and special sensory receptors that are very, very sophisticated and very complex. Further, the special senses are concentrated in our head. Everything, all 5 special senses are found in our head, whereas pain, for example, or touch is found everywhere. So those are the special senses that we're looking at today. As opposed to the general senses, which we've talked about before: touch, pressure, pain, temperature, proprioception, visceral. They're very simple structures. They are either free nerve endings, excuse me, or encapsulated nerve endings. But they are very, very simple structures. They're a dendrite. They're the end of a dendrite being stimulated. That's all they are. So let's take a look at olfactory receptors in our nose. The olfactory receptors are going to allow us to detect specific odors when the olfactory layer is stimulated. Here we go. Okay. Now, we have up to 100 million or so olfactory receptors inside our nasal cavity. They are located at the anterior surface, essentially the roof of the nasal cavity. There are what we call olfactory epithelium cells. They're simple epithelial cells designed to function as a chemoreceptor. That's all they are, because really an olfactory receptor is a chemoreceptor designed to pick up the presence of a specific molecule. And so in this case, the olfactory receptor is a bipolar neuron. We talked about that already in lecture today. A bipolar neuron has one dendrite and one axon. And in this case, the bipolar neuron has cilia attached on the surface of the neuron. And the neuron is embedded in a layer of mucus. And the cilia that are attached to the neuron are going to function much like the pseudostratified ciliated columnar epithelial cells do in the trachea, and that's moves the mucus around so we can swallow it. But the mucus is designed to allow chemicals, odorants, to dissolve in that mucus and be detected by the sensory receptors. They are nothing more than chemoreceptors. It takes a chemical molecule to dissolve in the mucus to activate the receptor in these bipolar neurons. And we can pick up about 10,000 different odors. And the signals then are sent up through the roof of the nasal cavity, up through the crista galli, up into the brain, into the olfactory nerve. So, and this is the region of the nasal cavity we're talking about. We can see that the olfactory epithelial cells line this area here of the roof of the nasal cavity, the anterior surface, if you will. Here we have the crista galli and the cribriform plate of the ethmoid bone, because the crista galli and the cribriform plate are together, and so the cribriform plate sticks up, allows the meninges to attach, and the crista galli sticks up, and the cribriform plate surrounds it. The cribriform plate is perforated to allow olfactory nerves branches to penetrate up into the brain, but it is the bipolar neuron located on the anterior surface of the nasal cavity that's going to get stimulated. And here we see this is an olfactory receptor here. It is a bipolar neuron. There's its dendrite, there's its axon, there's its cell body, and we have cilia along the surface here. Here is a essentially like a goblet cell secreting mucus along in here, and the molecules are trapped in the mucus and dissolve and are picked up by the olfactory receptor. Here's one, and here's another one, and here's another one, and all these little blue structures are the olfactory receptors. They are all bipolar neurons. And you can see now the branches of the olfactory nerve pass through the cribriform plate. There's little perforations there, and synapse in what a structure known as the olfactory bulb, which is found in the frontal lobe of the brain. And that sends signals down the olfactory nerve, but it becomes the olfactory tract at some point. So, because here's the frontal lobe right here, there's the olfactory tract, there's the olfactory nerve coming up through here, and at some point it becomes the olfactory tract right there. And if we're looking at our model here, let me show you this. And let's get here we have, here is our head model right here. And we're looking into the nasal cavity right here. Here's the nostrils here. This is the inside of the nasal cavity, and the anterior surface of the nasal cavity is located right here. So our olfactory receptors are located here. You'll notice close proximity to the tongue. There's the tongue right there. This is the oral cavity down here, and here's the nasal cavity here. And they're both open together. The pharynx, our throat, extends all the way up to the nasopharynx here and continues down into the oral cavity down here. We depend a lot on, and there's the nasal cavity, there's the oral cavity. We depend on odors a great deal in our ability to taste food. the odors given off by the food help us to identify the food, identify the flavor of the food. So when we have, consider if you've ever, whenever you have a head cold and you're not real hungry because food doesn't taste right, it just sort of bland because you can't smell the food. And that's because we depend on these olfactory receptors right up in here to tell us what it tastes like, in addition to the gustatory taste buds found down here on the tongue. So anyway. Let's go back to, so there's our olfactory receptors. The olfactory receptors can pick up to 10,000 different odors. Up to 10,000 different odors. We can identify them. We store the information in the portion of our brain that processes odors. And so when we are triggered by the same response again, we can identify it. Remember though that the olfactory receptor has no idea what's stimulating it. It's only when we start processing the information can we tell it's a rose or it's pizza or it is baking bread or roast turkey or a hyacinth, very distinctive flower odor, or like I said, roses, something like that. It's only when we process the information can we tell what it is. But it all depends on the presence of these olfactory receptors in the anterior surface of the nasal cavity. We have all these bipolar neurons. And again, you can see the bipolar neuron. There's the dendrite, there's the axon, and it leads to the openings in the cribriform plate. We are olfactory nerve here, and we are olfactory tract by the time we're up to here. And we'll see on the brain, on the brain that we dissect, that the olfactory bulb, that this area where we're receiving information on, is quite prominent. We're using sheep brains, and the sheep brain has a very large olfactory bulb, much larger than a human one. Okay, now, so that's olfaction. That is our ability to detect odors, and we store those odors in our brain. We recognize the odor. As we age, our ability to detect odors fades. By the time we're 50, we've probably lost a pretty fair number, maybe even 50% of our odor receptors and our taste receptors, and it continues as we age past that. Many elderly people suffer from malnutrition because they state that food doesn't taste good anymore, and so they don't eat because there's a lack of taste or a perceived lack of taste in the food. The food still tastes the same, but there's olfactory reception and their taste buds no longer can detect the flavors and the nuances and the subtleties in here. So it becomes rather bland. And so when food doesn't taste good, lots of older people don't eat because they have a lack of interest in eating, because they perceive to have no taste there. Or they use excessive seasonings. They may use lots of salt or lots of flavoring agents to try and recapture the taste. And there's lots of complications with that too. Now, remember that olfaction uses chemoreceptors. So does taste. Gustation, again, requires that the molecule, the flavor molecule, the chemical that's causing the flavoring in the food, whether it's a natural chemical or a synthetic chemical, has to dissolve in saliva to stimulate the chemoreceptors that are found lining the surface of our tongue. The only way we can taste food is to dissolve those food particles, release the chemicals into saliva so that we can determine what the flavors are. And then our brain will tell us what we're eating. Our eyeballs say, oh, look, it's pizza. It's pepperoni pizza. You expect pepperoni pizza to taste a certain way. You're used to eating pepperoni pizza, so you expect a certain flavor when you bite into it. You expect chocolate ice cream to have a certain flavor to it. But a fair amount of that ability to detect the flavors of the food depends on the odors. The molecules that are being given off from the food that you're eating, the odors that are coming up off the food are detected in your nasal cavity, since it's right there, right above your oral cavity, and your nose is right above your mouth, obviously. A fair amount of sensation on the flavor of the food is based on odors. And if we lose the ability to detect those odors, then we lose a large portion of our ability to sense flavors. Again, the analogy of if you have a head cold and you're congested, food may not taste the same. It may taste bland to you or have no flavor because you're not picking up the odors. So in either case, it depends on chemoreceptors to detect the food molecules, the food chemistry, the chemical molecules of the food going, triggering in the case of olfaction, the bipolar neurons. Now, in our mouth, we can detect 5 different categories of taste. 5 different categories of taste. We have, of course, the chemicals that are in the food have to be dissolved in here in our mouth. We can pick up the odors, the flavors of sweet, salty, bitter, sour, and what's called umami. It is what we call savory. It is the taste of protein. It is what you would detect, for example, when you're eating a freshly grilled piece of meat. You pick up that savory sensation in here, the protein flavor, if you will. This was not discovered until the 1980s. So this is a recent addition. It used to be sweet, sour, salty, and bitter, but now umami is the most recent of them. It is the 5th taste flavor that we can pick up. We mostly pick up our tastes within the taste buds that are found on the tongue, but we also have taste receptors on the soft palate and also on our epiglottis. Our epiglottis is a flap that will fold over and close off the glottis, the opening to the airway. So normally we have stratified squamous epithelial cells here because we eat, and we're going to, the movement of food through our mouth is going to tear away cells. That's what they do. So we have stratified squamous epithelial cells there to protect us. Now, within those stratified squamous cells, we also have the taste buds. These little buds are called papillae. They stick up and they are responsive to the 5 different flavors. Here we see what the papillae look like. These are little raised papillae here on the surface of the tongue, and they detect the various flavors. Here's what a taste bud looks like. There's our tongue. There is a pore leading to a papillae right there, and the chemical is brought in here, and we detect it inside this, and it sends a signal to the brain, and the brain tells us what we've detected. Is it sweet? Is it sour? Is it salty? Is it bitter? Is it umami? And many flavors are a combination of flavors. Many foods give us many sensations at the same time. Consider salted caramel. We're getting sweet and we're getting salt at the same time. You know, it's just a combination. You know, if you're eating barbecue, you may pick up the flavor of the sauce, as well as the flavor of the protein, as well as you would have the sweetness of the sauce, the peppery flavor of the sauce. It just depends. Maybe the garlic. You pick up all different sensations here when you eat. We just don't have one area that only gets sugary, sweet flavors and nothing else. So it has been said repeatedly over the years that there are areas of our tongue that only detect sweet and sour and salty and bitter. And that's really a misconception here, because if we only had isolated areas on our tongue where we would pick up sweet, for example, or bitter, or salty, or sour, then once food moved past that area, we wouldn't pick it up again. But when you eat something, you pick up flavors from throughout your mouth. We tend to have all the different sensations scattered throughout our tongue and our epiglottis and our soft palate. Because, where in the food process of eating does it say we can only pick up sugar here or only pick up sour here? So these taste buds are scattered randomly through and they suffer the same problem that olfactory reception does. They will wear out over time, and we'll lose about half of our taste buds by the age of 50. And then the rest will decline gradually over time. And in addition to the olfactory receptors wearing out and not functioning anymore, so will the taste buds. And we see this as a real issue in, like I said, in your elderly patients, because they will no longer be able to smell or taste their food, or not very well anyway. And so that causes them to lose interest in their eating. So malnutrition can play a real effect on the health of your older patients, because if food doesn't taste like anything, your patient loses interest in what they're eating. And it does happen. It happens quite a lot. So now let's move on here. Okay, so those are olfaction and taste. Those are two of the special senses. Now the next one is arguably the most important of the special senses, and that is vision. So we're going to take a look at the human eye. The human eye is probably the most complex of all of the special sense organs, located in the eye socket on each side of the nose. It's designed to pick up light rays and convert them into action potentials so that our brain can interpret what we're seeing. The eyeball has no idea what we're looking at. It just gets stimulated. So now, the outer surface of the eye is covered with a layer of connective tissue called the sclera. It is the white of the eye. In the front of the eye, there is a membrane that covers the sclera, called the conjunctiva. If you've ever had conjunctivitis, which is an inflammation of this membrane, another term for conjunctivitis, I think, is pink eye. If you've ever had that, it is an inflammation of that membrane that's covering the sclera. In the front of the eyeball, we have a clear layer called the cornea. And the cornea is where light passes through. The sclera is transparent at that point. We have the cornea. Light passes through the cornea. It goes through the pupil, the opening in the front of the eye, and strikes the lens. And we see that here is what our eye looks like. Now this is the blue layer there, is the white of the eye. In fact, let me show you on this model here. Back this up just a little bit. This is a huge eyeball here. Now, okay, here's our eyeball. It is not to scale, I can tell you that. So this white layer that goes all the way around is the sclera. This is the white of the eye. This large structure back here is the optic nerve leaving the eye. This is the cornea, the transparent portion here. Now the conjunctiva is covering the whole portion of the eye that's exposed, the part that we see. You know, the sclera that we see, the white part of our eyeball, is covered with the conjunctiva. The conjunctiva stops when we get to the cornea. Now, if you look carefully, here now, so I can take this apart without too much upset. You can see right in the middle here is the pupil. Light comes in through the transparent cornea. You can see it is transparent right there. You can see right through it. And the light will come in through the cornea and go into the through the pupil here. The pupil, let's see if I can get the cornea to come off. There we go. There is this area here that I just took off. This is called the iris. The iris is the pigmented portion of our eyes. It makes our eyes brown or blue or green or black or hazel or whatever. And the iris, the pigmented portion, is what determines the size of the pupil. When they contract, the pupil will shrink. And when they relax, the pupil will enlarge. And so the light will come in through this opening here. This is the pupil, and then the iris surrounds it. And underneath that is what we call the lens. This is our lens right here that focuses light on the retina. Now let's get back to this structure here. Now this shows you what, two-dimensional, but here is the sclera, the white layer. There's the iris. It's hard to see here, but the iris is at the pigmented layer. It is made of smooth muscle that will constrict or relax, and that determines the opening of the pupil here for light to come in. The cornea is this transparent covering over the front of the eye, and the conjunctiva covered this area along here and along here all the way around, but not where the cornea is located. This structure here is the lens. Light rays will come in here through the pupil, through the cornea, through the pupil, into the lens, and be bent and focused on the retina. The inner layer of the eye is called the retina, and the retina is where all the photoreceptors are located. So now so our ability to see depends on the movement of light rays coming in through the cornea and then focused by the lens onto the retina itself. Now, there are photoreceptors on the retina called rods and cones. The rods are going to respond to shades of gray and dim light, and the cones are going to deal with color. And color and visual acuity. We are more sensitive to color vision and detail with the cones, the cone photoreceptors. The rod photoreceptors allow us to pick up movement and function very well in dim light. However, all they do is generate an action potential. These bipolar neurons can only generate action potentials if we hit threshold. Just like we talked about in lecture today, the stimulus of the light rays have to be sufficient to trigger, to reach threshold in the cell body of the bipolar neuron. If we hit threshold, we get an action potential that goes to the brain. The brain still has to interpret what we're seeing. So, but what the lens does, and the cornea does a little bit, is bend the light rays. The lens bends the light rays so that we can focus the image that we're looking at onto the retina, on the photoreceptors. Now, the shape of the lens, we can alter that. It's called accommodation. And there's a muscle that attaches to the lens itself called the ciliary muscle. And so we can slightly alter the shape of the lens to accommodate objects we're looking at so we can get them in focus better. Okay, as I said, the iris, the pigmented portion of the eye, it allows light to come in. In dim light, the pupil gets bigger because the iris, which is made of smooth muscle, will relax. It will relax. When it relaxes, it enlarges the pupil, and so we let more light in. In dim light, we want to let in more light so we can see as best as we can. In bright light, our pupil will constrict. Smooth muscles of the iris will contract, and the pupil will get smaller. And so we can change the shape of the pupil by contracting or relaxing the iris. We don't need, in bright light, we don't need a lot of light to see well. So we restrict the amount of light coming in by constricting the iris. The pupil shrinks in bright light. In dim light, we need light. We need a lot of light, so the smooth muscles relax and they expand, and the iris gets bigger and the pupil enlarges. So in dim light, we'll have a large pupil. Like at night, you're outside walking around, and your pupil is very big because you want to see. You want to see better. You want to gather as much light as you can. In bright light, your iris contracts and your pupil shrinks because we don't need all the bright light. We only use what we need. So we restrict the amount of light coming in because we have more than enough light in a classroom. Now, once we get through the pupil, then the light rays will pass through the lens, and the lens will bend those rays to focus them onto the retina itself. And the ciliary muscles which surround the lens, I'll show you them when we start dissecting a lens, an eyeball. The ciliary muscles will make the lens thicken if we're going to focus on close objects, and allow the lens to become thinner to look at distant objects. So we actually have the ability to shape our lens from a thicker lens to a thinner lens, depending on, are we looking at something close up or are we looking at something in the distance? Because even though our lens is a crystalline structure that's transparent, it still has flexibility. So the retina is where we find our photoreceptors, the rods and the cones, as well as the blood supply that we depend on. Now, when you have an eye exam, one of the things that the ophthalmologist will look at is your retina and look at the capillaries supplying the retina. Are they functioning well? Are they clotted? Are they distorted? Or do they look like they're functioning normally? Because there are many, many health conditions that affect the retina. You know, a person with diabetes, diabetes mellitus, what your patients will often refer to as sugar diabetes. One of the first things that are affected by diabetes is an impairment of peripheral circulation, meaning that blood vessels constrict and reduce their flow in the periphery of our body. That includes the retina. If we start reducing the blood supply to the retina, the photoreceptors start to die. We don't like that. So Now, in our retina, though, let's talk about a healthy retina. The most sensitive portion of the retina is an area called the fovea centralis. In the fovea centralis, that is the area of the greatest visual acuity. And so we call this the fovea centralis. The fovea centralis, all cones, all color receptors. We have our greatest visual acuity located in the fovea centralis. So we get a highly detailed, our highly detailed images occur when light rays strike the fovea centralis. So let's take a look here. Now this is a cross section of our retina. We're looking here at our retina. Light rays come in this direction. And this is the outer surface of the retina. Light comes in this way. And we have the three layers of the eyeball. We have the retina. We have a middle layer called the choroid coat. And we have actually the sclera out here. This last layer is called the sclera. Now, the light comes in here. Our bipolar neurons are located here. And our rods and cones are located here. So light comes in this way. And it turns out this is a pretty good design because as the rods and cones become stimulated, they tend to warm up. And where they're located underneath the bipolar neurons and the ganglion, they can dissipate that heat directly into the bipolar neurons or directly into the choroid layer. But if they were found near the surface here, if the rods and cones were first instead of last, then a lot of that heat would not be dissipated properly, and it may likely affect our vision. But because the rods and cones are in where they are, the heat energy released as the photoreceptors are stimulated is easily dissipated. So just a little factoid. So anyway, rods and cones, the photoreceptors. The rods are on the periphery of our retina. They give us great peripheral vision. You're driving down the road and suddenly a deer darts out or starts to dart out on the side of the road, but you see it, as we say, out of the corner of our eye. Well, our eyes don't really have corners because they're round, but we say that out of the corner of our eye, we caught sight of a deer and we were able to respond because we saw it. Peripheral vision is based on our rods. The night vision is based on our rods. We don't see very well at night, but we do detect movement very easily. And again, our peripheral vision and our rods are going to allow us to see things all in gray, but we can pick up movement, we can pick up peripheral action very nicely. And again, they are located around the edges of the retina. Now the cones are color vision. They are located in the center of the retina. In fact, the fovea centralis, as I said, is the area where we only have cones. In bright light, we do very well for visual acuity at the fovea centralis. That is our area. As I said, it's our area of visual acuity. There is one spot, though, in the retina where there are no photoreceptors, no rods and no cones. We have an area called the optic disc or blind spot. That's where the optic nerve is leaving the retina. There are no photoreceptors at the blind spot. And so light rays that strike there are not detected because light rays will strike our blind spot. And we've often experienced that. How often have you been, driving and you see something and then you glance again and it's not there, or you look and there's nothing there and you look again and there's a car there. Because for that one split second you looked, the light rays of that car hit your blind spot. You didn't see it. And when you glance again, there it is. We've all experienced that. It's because of the light rays hitting the area we call the blind spot in the retina. Okay, so these are the rods and cones. We call them that because of their shape. This is the cone. It's shaped sort of like an ice cream cone. That's why it's called the cone. And this is the color vision. This is the visual acuity. Over here is the rod. Again, because of this shape here, this structure like a rod. This is going to be at the periphery of the retina and deal with movement and night vision and peripheral vision. So here is our structure within the retina itself. There's the ganglion connecting all the cells. Here we have the bipolar cell. Here we have the rods and cones right here. And so we stimulate the rod, stimulate the green rod. They're not really green. Stimulate the rod or stimulate the cone. The signal then passes, from the axon to the dent. It's detected here at we have one axon and one dendrite. This is the dendrite. This is the axon. We connect that from the receptor, pass through to the ganglion, which is going to lead us to collect, all the impulses from all the photoreceptors and take them to the optic nerve. Because light comes in here and it goes all the way to the inside lining in here where we have the epithelial, the visual epithelial layer, if you will, where pigment is located. And we have our rods, we have our cones. They're stimulated, and then they send the signal back through the bipolar neuron to the ganglion. And that leads to the optic nerve, because the optic nerve axons are down here. So you see, they don't show up here. So light comes in this way. It stimulates the end of the rods and cones. It sort of sounds like it's backwards. And from a construction perspective, it is backwards, but it's the way it works. And it works very, very well for us. Now, moving on. Our eyeballs have the ability to adapt to light and dark. When we go from dark to light, we adapt very quickly, because when we go from a dark area to a light area, we're stimulating all those cones, and particularly the fovea centralis, where we have all the greatest amount of visual acuity, and they kick into play very, very quickly. The cones regenerate rapidly. In dim light, the cones aren't going to work, but they regain their ability to function in bright light very quickly. When we go from bright light to a dark area, we go from inside our house and go outside at night. We adapt slower. To go from bright light to darkness takes more time. The rods will become activated in dim light slowly. There's an optic pigment called rhodopsin that shows up in the rods that takes time to regenerate, to show up, to become functional. And that's when we go from light to dark. The rods take more time to develop the rhodopsin. And so when we go from a brightly lit room to a dark room, or do from inside to outside, it takes a while to recover our night vision in here, because bright light destroys our night vision. It takes time, maybe 20 minutes or so, for our rhodopsin to come back. Whereas going from outside to inside, our eyes adapt very, very quickly. So our pupils also respond to light. In addition to allowing light rays to pass through into the the lens, the pupils will respond to bright light and dim light to regulate the amount of light that is striking the surface of the retina. There's only a set amount that we need. Now, on bright light, like in a classroom, the pupils are going to constrict. They're going to contract, and this is a parasympathetic response. They're going to contract, and they're going to limit the amount of light striking the retina. We only let in enough light that we need, not anymore. If we are in normal light, outside daylight, or maybe in a hallway or something where the light isn't as bright, our pupil enlarges, but not a lot. Again, just enough light to come in to do its job, allow the retina to do its job. The pupil will enlarge. We go from bright light to what we would consider normal light. Now, if we go into a dark room, then our pupils will open up even more to try and maximize the amount of available light that can strike the retina. And so the pupil, the shape, the size of the pupil is based on the contraction and relaxation of the iris, of the smooth muscles of the iris. So in dim light, our pupil gets really big, and in bright light, it gets very small. So now, I mentioned the blind spot. The blind spot is the area in our retina where there are no photoreceptors. There are no photoreceptors there. We call this the optic disc, or the blind spot. We call this the optic disc, or the blind spot. There are no photoreceptors here, so any light rays that strike the blind spot can't be detected. Now, there's a simple test that we do to take a look at the blind, to find where our blind spot is. If you look at you see on the slide, you'll see a black spot and a plus. If you cover one eye and you focus, it's hard to do on the computer screen, but I usually in class, I have these little pieces of cardboard with a plus and a black spot here. And if you cover one eye and bring the object close to you, bring the card close to you and focus on the plus. So you have the card in your hand, you've closed your left eye, your right eye is looking at the plus, and you bring the card closer and closer to your eye that's open. At some point, the black spot will disappear because the light rays bouncing off the black spot will have hit your blind spot. The black circle will disappear as you bring the card closer to your eye because the light rays are coming off of there. They hit your blind spot on the optic disc. Don't see anything. And you move the card back, it's visible again. You move it forward, it's visible again. But that one spot where those light rays are coming off of that black circle are not being detected. And then you switch to the other eye, you can do it again. And so the blind spot is there. It's a real thing. It's based on the presence of, or the absence of photoreceptors on the retina. So now within the eyeball, we've just looked at the retina and we looked at the lens, but within the eyeball, we've got what is the lens made of? The lens is made of proteins that are transparent. They are transparent. They are crystal clear. We call them crystallins, and it is a transparent structure. The lens is going to give us divide the eye into an anterior chamber and a posterior chamber. The anterior chamber is divided again into another anterior and posterior regions filled with a fluid called the aqueous humor. The posterior chamber is going to be filled with a transparent jelly. The posterior cavity is going to be filled with this transparent jelly that gives us the shape of the eyeball. It fills up this the eyeball itself. The eyeball is round because of this jelly inside here. The jelly is transparent, and it's easily seen when we dissect an eyeball. The jelly just sort of pops out for us. But the anterior chamber, the anterior cavity filled with aqueous humor. The aqueous humor is fluid that is accumulating there from the lacrimal gland. Now, we drain excess fluid through our lacrimal gland. If we don't, then the anterior cavity can fill up, and we can increase the pressure on our eye by having more and more fluid being retained in the anterior cavity. This is the whole basis of the condition known as glaucoma. Glaucoma causes fluid retention to occur in the anterior cavity, puts pressure on the photoreceptors in the eye, and gradually and slowly destroys them, destroying the visual field. So whenever you have an eye test, you go to your eye doctor, you get it's your eye examination time, and they do that little puff of air. You're looking in the thing, and it's okay, now a little puff of air now, and they hit you in the face on the eyeball with a little puff of air. That measures the pressure on the surface of the eyeball, and the pressure is based on the accumulation of fluid in the anterior cavity. That's your glaucoma test. If pressure is increasing, it's because the aqueous fluid that fills that up, what we call the aqueous humor, is not draining out properly through the lacrimal gland. Now, the posterior chamber, it's like jelly. It really is. Yeah. Okay, looking at the lens, this is the lens down here. We can see the anterior cavity up here, and this is the posterior cavity or posterior chamber behind us. There's the cornea right there. There's the iris right there. There's our lens. There is the edge of the conjunctiva, the protective layer that adheres to the surface of the sclera. And that's what gets inflamed during conjunctivitis. It doesn't cover the cornea. The conjunctiva is, inflammation of that is what we call pink eye. Now, this is what's left of the ciliary body here. The ciliary body helps to change the shape of the lens for accommodation. Now, this chamber in here, this anterior chamber, is filled with fluid, lacrimal fluid, draining from the lacrimal, formed in here. It's the same fluid we use to rinse off the surface of the eye every time we blink. If we don't release enough of that fluid, it accumulates in this chamber here, the anterior portion of the eye, and causes pressure on the retina. So, and there's the anterior chamber right there. Okay. And oops, and there's the vitreous chamber back here, where we have what we call the vitreous humor. So now this is the optic nerve. Here we can see the retina. You can see the ganglia layer out here, and you can see where the photoreceptors are located here, and the bipolar neurons are right here. So there's our blind spot. There's the retina on either side of the blind spot, and there's the optic nerve. Now let me go back here. I want to show you back up a couple pictures here. See what the retina looks like. There. Now see, this is the retina here. The ganglion neurons that are going to leave, these are going to form the optic nerve. There are the bipolar neurons right here, and down here are the rods and cones, and this is the choroid layer. So if we go from here back up to here, we can see that our rods and cone layers are right here. The rods and cones are there. And the bipolar neurons are right here. And this is the ganglion that's going to lead, that's going to form these are the axons of the optic nerve right here, and that's the optic nerve. So anyway, blind spot, no photoreceptors, retina, and there's the optic nerve. Now, something that occurs to us when we look at images, of course, is we have to bend light rays. We refract. The bending of light rays is called refraction. The cornea and the lens bend the light rays. Now, sometimes that doesn't work well for us. A good, healthy lens will bend the light rays and focus them directly on the retina. But sometimes it doesn't work that way. Sometimes we bend the light rays, but the focal point, the point where the image should be in focus, is not on the retina, it's in front of the retina. Individuals that are nearsighted will take an object, they're looking at something, a picture, a text, a book, and they'll bring the object in very, very close to read it. They have what's known as myopia. They're nearsighted. They can see things very well up close, but they can't see well in the distance. See, and what's happening is that their lenses are focusing are generating what we call the focal point, the point where all the light rays converge and the image is in focus. They're generating a focal point in front of the retina. So the short-term fix is to bring the object in closer and essentially move the focal point from in the middle of the eyeball to the retina itself. So a person with myopia or that's nearsighted has great visual acuity up close, but they'll often bring things up close to their eye so they can see it better because it focuses then on the retina. Now we fix that with corrective lenses. We use concave lenses, lenses that have a depression in them. Now, the other side of this is called hyperopia. This is farsightedness. The image here is being distorted by the lens, and the focal point is actually behind the retina. Now, we don't pass behind the retina. The light rays don't pass behind the retina. But what the light rays do is that when they're striking the retina, they're blurred. The focal point would be theoretically behind it. So a symptom of developing farsightedness is when you're reading something is to have to hold it further and further away from your eyes because you're moving the focal point from a theoretical behind the retina to onto the retina. And so these people that have hyperopia see distant objects very, very clearly. But up close, they can't see anything. So they have to use corrective lenses also, and they use what are known as convex lenses. The lenses bend out instead of bending in. Myopia and hyperopia. Here we have normal vision. The object is focused directly on the retina. With myopia, the focal point is in front of the retina. And one of the symptoms of this is when an individual will bring an object they're looking at up close. The closer they get to the eye, then this object moves from here. The focal point moves from the middle of the eyeball to the retina. A person that's farsighted will have the focal point. Now, there's normal vision with the focal point on the retina, but a person who is farsighted, the focal point is behind the retina. What's hitting the retina is blurred vision. They will often move the object away from them, further away, to bring this focal point to the retina. It's often called presbyopia too, or hyperopia. You're either nearsighted, you can see things very well up close. You're farsighted, you can see things very well far away, but not up close. So, person who is colorblind, the person who is colorblind has a problem with their rods and cones. It is a problem of not having the right kind of cones. The cones that detect red-green colors are absent or non-functional. Red-green color blindness is the most common type of color blindness. It is usually shows up as a sex-linked trait, a recessive trait passed on from two male offspring from their mothers. Most males are the most commonly have red-green color blindness, and it's the gene for the color blindness is carried by their mother and passed on to the male offspring. Now, another condition, night blindness. Night blindness affects the ability to produce an adequate amount of rhodopsin in the rods. Rhodopsin is often caused by an absence of vitamin A. And when we were kids, we were always told to eat our carrots so we could see better. We eat our carrots so we get vitamin A. You know, and they say rabbits eat lots of carrots and they see well at night. We've all heard that story. Well, there is some truth to it. Vitamin A contains, carotene found in carrots has high levels of vitamin A. And if we can maintain, if we have adequate amounts of vitamin A, we are going to generate lots of rhodopsin, and we can see as well as we can in dim light. The rods will be at their peak effectiveness. If we don't have a lot of vitamin A, then we will have night blindness and we will have very poor night vision. So, now the test for color blindness involves a series of test plates here that are designed to let you see if, this is like detecting red-green color blindness. A plate will have a series of bubbles on here of usually like a light, in this case, a light green color with the orange bubbles showing the signal, the number 25. If you have a type of color blindness, you would see everything as a shade of gray. Same here. We see more greens of different shades, and we see different shades of red. We see the number is 56. If there's no indication of color blindness, particularly red-green, you would see the number 56. Here, it gets a little trickier. You have a 6 embedded in here. Again, if you have no red-green color blindness, you would be able to see the 6. If you didn't, in both of these other cases, you would see it as shades of gray. There's a 29. There's a 45. And there's an 8. In all cases, though, a person with very red-green color blindness would see these as shades of gray and likely wouldn't detect the numbers in here. So this is what, a very simple book of color plates. We use the computer now, but the original work was done years ago in Japan and by an ophthalmologist named Ishihara. And he's based the ability to detect, and it still works, to detect color blindness based on the ability to perceive the numbers or not perceive the numbers. So these are two tests. The Snellen eye chart is the chart that allows you to determine visual acuity. We've often heard, your vision is 20/20. Well, what does 20/20 mean? Well, this chart is designed to take into account, the Snellen eye chart takes into account light rays bouncing off these different rows of letters. And this line down here is marked as the 20-foot line. So this test means that with the Snellen eye chart, if you have an individual that stands 20 feet away from that eye chart, mounted on the wall at 6 feet above the floor, if they close one eye, and their vision is perfectly normal, they should be able to read what we call the 20-foot line while standing 20 feet away and read all 6 letters unaided, no glasses, no corrected lenses, no contacts. So 20 feet away, you can read the 20-foot line. And so if you can, then your vision is considered 20/20, reading the 20-foot line 20 feet away. Now, if you can read the line below that at 20 feet away, the 6 letters there, then your vision, that's the 15-foot line. And your vision is considered 20/15, meaning you can read at 20 feet what most people can only read at 15 feet, meaning your 20/15 is very good vision. You're seeing things at 20 feet away that most people have to move in to the 15-foot line. On the other hand, if you look at this, if you're standing on the at the 20-foot line and you can only see this large letter E, that's the 200-foot line. An individual with a healthy eye could read this letter while standing 200 feet away from the chart. But if the only thing you can see is the letter E while you're standing 20 feet, the only thing you can see is the letter E when you're standing 20 feet away, then your vision is what we say 2200. You see at 20 feet, but everybody else sees at 200 feet. So this is a very simple test for visual acuity. It's been around for years and years. This is essentially what we now see when you're looking through that viewer that the optometrist sets up, and they keep changing the lenses around. They go, now this or this or this or this, which is, it's a improved version of the Snellen eye chart. The other chart here is the test for astigmatism. Astigmatism is caused by a blurring of images on the retina caused by the cornea. The cornea has thickened in some areas. And so we see some lines on this chart as thicker or darker, or thinner and lighter. Now, I will tell you that this is what the way the chart is drawn on here is how an individual would see it if they had astigmatism. Because I've had too many people look at that and say, oh my goodness, I see these lines as really dark and I see these lines as really light. Well, you do, because they are drawn that way. A person with astigmatism would see a thin line here and a thick line here. The lines at 9 and 3 on the chart are thicker because they're drawn that way. This is how an individual with astigmatism would see, instead of all the lines looking exactly the same, the damage to the cornea, the thickness of the cornea, is affecting their ability to have the light rays strike the retina properly. Now, fortunately, astigmatism is one of those things that we correct easily with glasses. So, see what's happening is our cornea with astigmatism, our cornea is, instead of focusing the light rays onto the vision, they're allowing it to spread out. So astigmatism gives us a blurred, altered image on the retina because the way the light rays spread. Instead of focusing it direct, to a point, they let them spread out. Okay. And I think this is a really good point to stop. We will pick up next week. We'll talk about the ears. So I want to play with the ear model. And then next week we will dissect. I will dissect for you. I'll go through the slides and then we'll do some dissecting so you can see what it really looks like. And then we'll be done with lab. So any questions on anything before I get us out of here? Okay, you may have noticed that the muscle test opened up today. It'll be open for a week, so you have plenty of time to get ready for it. We need to get that done. So if you have a problem with scheduling, let me know, and I'll be more than happy to give you a little more time to get it done. But it should have opened up today. So, and it won't be, remember there's only 60 muscles. So anybody have any questions on anything? Okay. Do you know when the lecture test opens for, obviously for bones and muscles? It won't open until after the lab test is done. So it'll open next week. So, and You're welcome. And since I haven't written it yet, I need to go ahead and do that. So maybe I'll write up while I'm watching basketball tonight. So, okay. Because I don't care either way. Okay. Let me get you out of here and I will see you all on Wednesday in lecture.